Hello students, once again welcome back to our biology class. Myself Dr. Raghavendra here. So, in the earlier class we studied about the structural units of these biomolecules, and then we entered into the concept as such, physiological importance of the biomolecules. And we began with the proteins as the important biomolecules. Why? Because proteins are the mainly structural proteins. They can be the receptors, they can be the hormones, they can be the enzymes, they can be the antibodies. In various ways, proteins are very, very essential and they are playing the vital role. So, it may be the defensive mechanism to the body or immunity by the immunoglobulins. It may be the homeostasis or proper metabolism, yes, mainly by the presence of the enzymes, right? So, we have discussed a variety of mainly the kind of proteins. The proteins which are very much abundant in the entire biosphere, it is rubisco. The abundant protein in the animal kingdom, it is collagen that we have discussed. The role of GLUT4, it is mainly glucose transportation, yes. And what is hypoglycemic hormone? It is the insulin that reduces the glucose level in the blood. So all these, yes, we have discussed. That is called as a physiological role of mainly the proteins. When we talk about the structural aspect as such, proteins here is mentioned polymer of the amino acids. So these can be amino acids. When it is called polymer, it can be called as heteropolymer, or it can be the homopolymer. So what we can say, the long chain of similar or the same amino acids. Okay, AA means amino acids. So it can be heteropolymer, long chain of, we can say that it's a long chain of different amino acids. So then it's called a heteropolymer. So how these amino acids are attached to one another? Let me tell you, considering a simple example. This is the carbon with its NH2 group, and then here it's a side chain R1, and then here is the carbon with O, as well as we can say that again it is the OH group. So this is one amino acid plus now addition of one more amino acid to this, NH2 carbon, the R2 group, and then one more carbon, double bond O, and then OH. So two amino acids, now they are getting combined. What will happen? So it is a kind of dehydration reaction, removal of the water group. So ultimately it leads to the formation of, let me tell you, NH2, the carbon, the R1, and then next carbon, double bond O, next is the nitrogen and one H group, and then what we find, again the carbon, the R2 group, then the carbon, double bond O and OH. So what happened here? The formation of, this is what we say the peptide bond. So formation of a peptide bond occurs. Peptide bond with the release of H2O. From where this H2O has come? Yes, mainly one H from the NH group and this OH group. So has removed and resulted into removal of the water or release of the water, and the reaction is a kind of dehydration reaction. Okay, so yes, removal of the water group or dehydration, you can say. So this is how actually the amino acids are attached to one another or linked to one another. So when we talk about the mainly protein structures, the protein structures can be as straight as linear. Say for example, amino acid 1, amino acid 2, amino acid 3, amino acid 4. So this is called as mainly the C terminal C terminal represents the last amino acid, and here it is called as N terminal. N terminal represents the first amino acid. Okay, so the long chain C and N, this is called as a linear structure of the protein. Linear structure is also called as a, we can say that, primary structure. So this is primary structure. Well, so it can be the other type also. What? It can be forming the loops or the helix or helices. So we call it the alpha helix. Alpha. Okay. So this is the C terminal, it is the N terminal, we call it as a alpha helix. Alpha helix is also called as a secondary structure. Secondary structure. So this various type of alpha helices, I hope this reaction you clearly understood how actually a peptide bond is formed. Yes, peptide bond is formed with the OH group of the previous amino acid and NH2 group of the next amino acid. With the release of the water, yes, peptide bond is formed. This I hope it is very clear. And now we are talking about if it is a homopolymer, all the amino acids are same kind. It may be the cysteine only, or methionine only. If it is a heteropolymer, yes, it has a serine, it has a cysteine, it has a methionine. Various type of amino acids are linked by what? Peptide bond. So that is called as a heteropolymer. Well, so this reaction is very clear now. Let us go ahead with what are the other different kinds of what we can say the structure of mainly the proteins. Let us understand that also. So when we talk about basically, yes, what you understood, if it is a straight or a long chain of amino acid, we call it primary structure. This is the primary structure. And if it is a helices or helixes, yes, it is called the secondary structure. So complexity keep increasing, it never decreases. So we can say that is a N end. Okay, yes. So it is a C terminal here, N terminal here, multiple helices we can say. It is called as a tertiary structure. Yes. So the third level is yes, tertiary structure. Tertiary structure refers to be mainly multiple helices clubbed together. Okay. So the tertiary structure also yes involves what we can say that there are the structures can be somewhere like this. So we call it as mainly the loops. So beta pleated sheets we can say. It can be yes mainly starting with the N terminal ending with the C terminal, or it can be yes tertiary structures. It can be like a woolen ball. Okay. So N terminal and C terminal. So it can be multiple kinds like this. So all these are called as mainly the tertiary structures. So we can say that there is a formation of bond. So intermolecular bonds are formed between these loops. Okay. So there is a intermolecular bond will form. We call it as a beta pleated sheets. Beta pleated sheets also forms the tertiary structure only. I hope you are understanding. So what is actually here we are observing. So mainly the linear chain structure is called the primary structure, and it is becoming the spirals. We call them as a helix, alpha helices we say, multiple. Or it can be much more complex helices interwoven like a woolen ball, or beta pleated structures having a bond between them itself. It is a tertiary structure. So, as such, to consider, imagine this is a polypeptide chain. Okay, it is a polypeptide chain. What you are observing. So, number of amino acids. So, we call these are the polypeptide chains. Well, so this is the polypeptide chain, means number of amino acids. Amino acids are here, here and here, everywhere. Okay. So we call them as polypeptide chains of amino acids. But these are again linked by, yes, particular bond between them. Okay, they are connected by the bonds between them. So this is also called as pleated structure. This is all known as beta pleated structure. Okay, next comes what we can say quaternary structure. The fourth important thing is quaternary structure. It is much more complex now. Here, quaternary structure we are going to understand. So, in the quaternary structure, what we can see mainly, the structure is so complex. So, it is forming the different units and clubbed together. So 2, 3, 4, 5, 6 units clubbed together. So these are the 2 alpha units or 2 alpha chains with a complexity and 2 beta chains with a much more complexity. So 2 alpha and 2 beta chains, totally 4 chains, and it is a structure of hemoglobin. So hemoglobin having 2 alpha and 2 beta chains. So it is called as the quaternary structure. So subunits, okay, are combined now. This is all about mainly the primary, secondary and quaternary structure, and in this we have discussed how the proteins are mainly made up of. They are made up of the linear structures, the amino acids. Amino acids are linked with one another, what? Mainly by the peptide bond, and then what we are observing, the amino acid, the first one is called the N terminal and last one is the C terminal. So length of the polypeptide chain, it may be 100, 1000, 10,000 also. So based on the higher molecular weight, more the amino acid, lesser the molecular weight, the smaller the protein or the lesser the polypeptide size. So when it forms the helices instead of a linear chain, the helices, then it's called the alpha helix secondary structure. These multiple helices are clubbed or they are going to forming the mainly the bond between each other, these long chains only, or it is going to form the much more compact tangled mass of a woolen ball, like that it forms, then it is a tertiary structure. So such complex tertiary structures, so we call them as alpha units or beta units. So they club together 2 alpha 2 beta. Best example is hemoglobin that is seen in the RBC, the oxygen or carbon dioxide carrying mainly the protein, we can say that. So this is what we say the quaternary structure. So this is all about the proteins what we have studied. Let us understand now. When we talk about mainly, yes, we need to understand certain important functional aspects as well as the structural importance of mainly the what we can say the carbohydrates also. Let us study that. So proteins, we have studied the polymer of amino acid, but when we talk about carbohydrates, they are the polymer of the sugars. Okay, so yes, we call them as the polymer of sugars, and these also have lots of importance in the maintenance of the body physiology. Yes, we need the sugars or the glucose every now and then, because 30% of the glucose what is obtained from the nutrition or diet, that all utilized by the brain itself. Remaining 70% is left for the body for its maintenance. Okay. Now, when you are talking about mainly here the polysaccharides. So we can call them as they are basically the polymer of sugars. So polysaccharides can be heteropolymer, yes, as well as homopolymer. Heteropolymer and then homopolymer. Well, so when we say that heteropolymer means they are mainly made up of different types of sugars. So made up of yes, more than one type of sugar or different type of long chain, we can say types of sugars. So here these are made up of similar type of sugars, similar types of sugars. Okay, well, so looking into what are these heteropolymer? Yes, it can be glycogen, it can be inulin. Yes, sorry, these are mainly the heteropolymer as well as the homopolymers. Yes, we need to consider mainly here the heteropolymers such as yes, glucose amine, N-acetyl, it is N-acetyl, galactosamine, amine is also called as NAG. And then even we can say that cotton, also a type of what we can say it is a heteropolymer. Now when we talk about homopolymer, yes, it can be the glycogen, it can be the starch, it can be the inulin, it can be the cellulose. Okay, even what we can say chitin. Chitin also a homopolymer, yes. So why we call these are a heteropolymer? Because of having the different type of sugars and making a long chain, heteropolymer. So when you talk about mainly heteropolymer as well as a homopolymer, fine. So here glucosamine, N-acetyl galactosamine and even the cotton also yes can be a heteropolymer. Whereas the glycogen is basically made up of glucose only, lots of glucose combined together forming the glycogen. And glycogen is also called as animal starch. It is a reserve energy for the animal body. Starch is mainly seen in reserve nutrient or the reserve energy in the plants. Inulin is mainly the homopolymer of fructose. Okay, so inulin is mainly homopolymer of fructose. It is made up of. Then cellulose as well as yes, cellulose as well as chitin. They are all again having the glucose only as the units in them. So when I talk about a single, mainly what we can say the example. So as such, they can be so simple and they can be so complex also. So when I talk about the starch, starch is much more complex than that of the cellulose. Why? Because let me tell you, starch, large and complex, okay, uptakes the I2, okay, or iodine stain. Whereas cellulose, yes, cellulose seen in the cell wall of the plants. So it is mainly smaller compared to starch. And simple. Won't uptake, it won't uptake what we can say, yes, I2 or iodine stain. Yes, so it won't react with the iodine, whereas starch react with the iodine and gives a positive test. So this is what we talk about. Where we find the starch? Yes, you soak the grains overnight, dry them under sunlight. Yes, you will find a good amount of starch in that. Even the maida is having a good amount of starch. The sago or sabudana, we say lots of starch. Starch is very difficult for our digestion, right? We need to utilize the sugars which are easily digestible. Best is the fructose, the sugar present in the fruits. Fine. So starch is a large complex, uptakes the iodine stain, whereas cellulose is smaller compared to starch and simple, and it won't uptake the iodine well. So now, how actually these taking the simple example of glycogen. Glycogen as such we observe. So it is what we say mainly the N terminal. Sorry, it is mainly the non-reducing end, we say. Non-reducing. So this is a long chain, keep on growing. And here, yes, we find these are the reducing ends. Reducing end, non-reducing end. Okay? Fine. So, what actually the long chain, if we take this section, here, how actually they are made up of? The glucose, they are attached, the first and fourth carbon. So, glycosidic, glucose attached by glucose molecules attached by glycosidic bond, glycosidic bonds between the first and fourth carbon. I will show that. And here, yes, what actually they are, the side chains, right? So here we can say that the glycosidic bond, glycosidic, this is the glycogen what we are studying, glycosidic bond between the first and sixth carbon of the next. Okay, so let us understand how actually they are made up of. So when we are discussing about this, definitely in the exam also, yes, it will come as an important question. So here to show you this, so this is the sugar with its terminal, what you can say, oxygen, and here what we can find mainly the first, second, third, fourth. Here is the fifth carbon and sixth carbon is here. Then what happens? Glycosidic bond. Yes, you are observing the glycosidic bond. So here again, this is the sugar molecule what I am drawing. It is a hexose sugar. The terminal carbon, first carbon, second, third, fourth. Yes, here is the fifth and here is the sixth carbon. So glycosidic bond what you are observing in between the fourth of the previous and first carbon of the next. Okay, so first as well as the fourth carbon connected by mainly glycosidic bond. Now here the side chain, what we can say, reducing chain. Here is the sixth carbon, and then a glycosidic bond is here, and what you are observing, this is the side chain. Okay, again it happens like this. So this is what we say terminal oxygen, 1, 2, 3, Yes, it is the fourth. And again, what is here? Here it is mainly the first carbon and sixth carbon. What we can say? This is the glycosidic bond you are observing. So a question will come just like this, and they will give you the diagram, and simply the line diagram they will give, and you need to understand as well as you need to justify your answer. Yes, what is the non-reducing? It keep growing. Reducing, yes, it stops somewhere, right? How they are attached? So in the non-reducing glycosidic bond between the fourth carbon and first carbon of the adjacent sugars, and in the reducing end, yes, it is the sixth carbon and first carbon of the next sugar. So this you need to remember. So thus what we have studied here basically, it is all about mainly the polysaccharides, and then they can be what you can say the physiological importance, glycosamine, N-acetyl glycosamine, and cotton. So cotton is basically the polymer of the different kind of sugars, and then so here glycogen is polymer of sugar only, starch is a polymer of sugar only or glucose, inulin is a polymer of fructose, cellulose is mainly the cell wall, it forms chitin. Yes, it is exoskeleton of arthropods. Arthropods. So this is all about what we have studied regarding the physiological importance of basically what we call it as the polysaccharides. So in today's class we studied about basically how these proteins are made up of. Proteins are made up of what? Proteins are made up of amino acids. So long chain of amino acid, what it is called as? Long chain of amino acid is called as a polypeptide chain. So how the amino acids are get attached to one another? Yes, by losing the one water molecule. Yes, they form C double bond O and NH. This is a peptide bond between the adjacent amino acids. So thus they form the polypeptide bond, and then generally a protein is a polymer of the same amino acid, we call it the homopolymer, different amino acid heteropolymer. N terminal in the protein means it is a starting of the first amino acid, C terminal the last amino acid we say. So linear chain means a long chain of amino acid, no spirals also, it is called the primary. When spirals start forming, it is secondary. Multiple spirals or helices, then it is a tertiary. It may be much more tangled like a woolen ball. Yes, it is a tertiary. Yes, quaternary means multiple sets of these, what you can say, the tertiary units. Then it become a quaternary structure. Best example is a hemoglobin, 2 alpha, 2 beta we have studied. So polysaccharide, yes, we can say that they can be the homopolysaccharide, heteropolysaccharide. Homopolysaccharide is what? It is having a similar type of sugars. Heteropolysaccharide, different kind of sugars we can say. And generally, the sugars basically what we can say mainly the glucose. So the sugars can be like we have studied in the earlier class also, 3 carbon trioses, 4 carbon tetroses, 5 carbon pentoses, 6 carbon hexoses. Okay, glucose is a hexose, ribose sugar is a pentose that we have studied. So how actually the glucose to glucose they get attached by the glycosidic bond. So first and fourth carbon in the adjacent sugars, that is a mainly in the non-reducing chain, and non-reducing to reducing chain, how they get attached? Sixth carbon of non-reducing sugar to the first carbon of the reducing sugar. This we have discussed. You need to know all these, otherwise it will be very difficult. Fine. So we are done with almost everything. Where the glycogen? Yes, glycogen is also called as animal starch. Animal starch, which is present in the liver. Okay. So the glycogen we have taken here, a long chain of glycogen that can be exhibiting. So as such, if we consider glycogen, it can be forming like this. Lots of side chains we can find. Okay, so it is not so simple. It is a complex of what we can say, mainly the glucose. So it is a complex of the glucose. This is in the reality how it occurs. It present mainly abundantly stored in the liver as well as in the muscles, but when it is energy required, glycogen is broken into glucose. So the mechanism is called the glycogenolysis. If the glucose also together they combine in forming the glycogen, yes, glycogenesis we call it as. You need to remember these two terms also. What is glycogenolysis? Breaking up the glycogen. So glucose molecules combine and forming glycogen, glycogenesis. Okay. So this every now and then happening in our body. So that's all for the today's class, and in the next class we'll discuss regarding the details of the nucleic acids also. Till then, take care and study well. Thank you very much.